Meet Scott and Mark Kelly. They're identical twins. They also happen to be the only identical twins in history working as astronauts for the U.S. National Aeronautics and Space Administration, or NASA for short. NASA scientists decided to take advantage of this unique situation and embarked on the first twin study. The twin study was designed as a nature versus nurture study and evaluated the effects of space travel on the human body. Biological samples of each twin were compared before, during, and after Scott's one-year stay at the International Space Station. Mark, unfortunately, had to remain on Earth. NASA scientists found that spaceflight affected some of Scott's gene expression. While most of it returned to normal after landing, 7% did not. The NASA scientists think this may result in longer-term changes in genes related to, among other things, his immune system. His space travel resulted in hyperactive immune activity, indicated by a spike in certain cytokines and an increase in levels of some proteins known to help maintain insulin activity. Changes in his environment, including escaping gravity, led to a change in Scott's immune activity. Similarly, changes in our lifestyle, as described in the hygiene hypothesis, may explain changes in our immune activity, resulting in an increased incidence of allergic and autoimmune diseases. The hygiene hypothesis is only one piece of the puzzle that looks at the increase in allergic and autoimmune diseases. According to epidemiologic data, there was a clear decrease in infectious diseases in developed countries during the second half of the 20th century. Scientists think this was due to the establishment of the public health system, improved sanitation standards, vaccines, and use of antibiotics. During the same time period, there was an increase in the incidence of allergic and autoimmune diseases in several developed countries. The prevalence of asthma increased by 59% in Australian children from 1964 to 1990, and by 38% in U.S. children from 1980 to 2003. Likewise, the prevalence of multiple sclerosis, or MS, in Vestford, Norway almost tripled from 1963 to 2003. Could this decrease in infectious disease and concurrent increase in allergic and autoimmune diseases simply be coincidence? A deeper dive into the epidemiologic data shows some interesting findings. One finding is migration patterns. Studies show that when immigrants migrate as children, they acquire the same incidence of allergic and autoimmune diseases as the host country. Hammond and colleagues reported that in most regions of Australia, the prevalence of MS in migrants from the UK and Ireland in 1981 was significantly lower than in their native countries. More recently, investigators reporting on the prevalence of MS in Ontario, Canada between 1994 and 2014 found that, while the general prevalence almost doubled, prevalence in immigrants more than tripled, with the risk of MS being higher in those who had migrated prior to the age of 20. Another interesting finding from epidemiologic data are differences in socioeconomic conditions among countries or regions with a high versus low incidence of autoimmune diseases. This was best highlighted by a study comparing the populations of Finland and bordering Karelian Republic of Russia. Both populations belong to the same ethnic group and live in the same climate. But Finland belongs to a higher socioeconomic class than Russian Karelia. Researchers found that Finnish children had a 6 times higher incidence of type 1 diabetes and a 4.5 times higher incidence of celiac disease than children from Russian Karelia. What is the common thread among these findings? They point to some form of environmental factor as the culprit for the rise in allergic and autoimmune diseases. This is where the hygiene hypothesis comes in. The hygiene hypothesis was first proposed in 1989 by David Strachan. He looked at more than 17,000 British children born the same week in March 1958 and followed them for 23 years. Strachan reported an inverse relationship between the incidence of hay fever at 11 and 23 years of age and the number of children in the household at age 11, especially older children in the household. He concluded that the lower incidence of hay fever in large families may have been due to infection in early childhood, transmitted either by unhygienic contact with older siblings or infection acquired prenatally from a mother infected by contact with her older children. Strachan's finding of an inverse relationship between hay fever and number of siblings was recently replicated in a global study in over 500,000 children from 52 low and high-income countries, where a higher incidence of hay fever was significantly associated with more affluent countries. Others have also shown a relationship between living conditions and allergic diseases. Children are less likely to develop atopic dermatitis if they have 3 or more siblings, are exposed to pets, are sent to daycare, or live on a farm. Therefore, according to the hygiene hypothesis, as originally proposed, the recent rise in allergic and autoimmune diseases is due to lack of exposure to infectious agents. 2 other hypotheses have since expanded on this concept. One is the old friends hypothesis. Just as humans have co-evolved with gravity, and its absence can lead to changes in our immune system, we have also co-evolved with certain harmless organisms called old friends. These old friends include helminths, saprophytic microbacteria, and lactobacilli. Our old friends were not only tolerated by our bodies, but they also modified our immune systems through different mechanisms, such as amplification of B and T regulatory cells. Helminths are acquired through contaminated soil, food, and water. By moving to cities and establishing public health systems, we have lost contact with most helminths and no longer experience their immunomodulatory effects. Researchers speculate that this absence may contribute to the increase in autoimmune diseases, such as MS. Several epidemiologic studies, including the latest in 2008, have concluded that the increase in MS in Sardinia, Italy, over the last 4 decades is partly due to the eradication of P. falciparum malaria 10 years earlier. Scientists believe that the residents of Sardinia have genetic traits previously selected because they protected against P. falciparum by providing increased immune reactivity. In the absence of this pathogen, these genetic polymorphisms resulted in excessive inflammation and became risk factors for chronic inflammatory disorders. Another concept building on the hygiene hypothesis is the microflora hypothesis. This hypothesis proposes that changes in the human gut microbiome disrupt its immunomodulatory effects, leading to the development of allergic and autoimmune diseases. The gut microbiome of an average 70-kilogram adult is made up of 39 trillion bacteria, more than the total number of human cells. It also consists of about 2,000 species, not all of which are beneficial to the host. Bifidobacteria and lactobacilli are considered beneficial, while staphylococci and clostridia can be pathogenic. The gut microbiome is unique to each individual and changes throughout life in response to nutritional, physiological, and/or pathological events. This is clearly highlighted by the differences in the microbiome composition and diversity of populations around the world. Hunter-gatherer populations, such as the Hadza from Tanzania, have little or no access to medical care and suffer from multiple infections of gastrointestinal pathogens and parasites. Their diet consists mainly of starchy foods, wild game, and honey, and their gut microbiome is very high in diversity and enriched in Prevotella and other species. In contrast, Western industrialized populations live in good sanitary conditions and have access to medical care, including antibiotics. Their diets are high in protein and fats, and their gut microbiome is low in diversity, depleted in certain species such as Prevotella, but enriched in others like Bacteroides. The gut microbiome affects many physiological functions, including metabolism, cognitive development, and immune response. For instance, a Swedish study compared the postnatal intestinal colonization pattern of 24 infants born vaginally or by cesarean section. Those born by cesarean section had a lower total microbial diversity, lower diversity of the Bacteroides phylum, and decreased T helper 1 responses in the first 2 years of life. Does this disruption in immune response homeostasis due to a changing gut microbiome play a role in the recently observed increase in autoimmune diseases? According to some epidemiologic studies, the answer is yes. These studies suggest that diversity of microbial exposure acquired through living on farms, being breastfed, or receiving oligosaccharide prebiotics may protect against allergic diseases such as asthma or atopic dermatitis during childhood and adolescence. For autoimmune diseases, it has been reported that patients with MS have gut dysbiosis and a specific type of microbiome with a relatively low abundance of the Bacteroides, Parabacteroides, Prevotella, and Lactobacillus, and an increased abundance of other genera, including Bifidobacterium. Several mechanisms that explain how the microbiota impacts immune response in MS patients have been proposed, including activation of T-cell-mediated response, mainly of Th1 and Th17, and by altering the ratio of effector T cells versus T regulatory cells. A study of the microbiome of 34 monozygotic twin pairs discordant for MS provides more information. This study used a germ-free transgenic mouse model of experimental autoimmune encephalitis, or EAE for short. Researchers found that when mice were colonized with fecal matter from both sets of twins, microbiota derived from twins with MS have a different profile, induced a significantly higher incidence of EAE, and produced less IL-10 than microbiota derived from healthy twins. Similarly, treating mice colonized with fecal samples from healthy twins with anti-IL-10 neutralizing antibodies increased EAE incidence compared with those treated with isotope control antibodies. When Scott Kelly returned to Earth and started readapting to his old friend gravity, most of the observed biological changes returned to nearly his pre-flight status, while few persisted after 6 months. This begs the question, what if we bring our old friends back? Should we espouse a return to pre-industrialized living? Would adaptations in our microenvironment reduce our risk of developing autoimmune diseases? In fact, recent studies in mice have shown that intestinal helminths can change the composition of the gut microbiota in a way that may be beneficial. In humans, data in MS patients have shown similar results. A prospective double-cohort Argentinian study compared 12 patients with relapsing remitting MS, or RRMS for short, infected with a parasite to 12 healthy subjects and 12 uninfected patients with RRMS. All patients were followed for more than 4.5 years. As compared with uninfected patients, infected patients showed a significantly lower number of relapses, reported minimal changes in disability scores, and exhibited significantly lower MRI activity. Immunological evaluation showed that parasitic infection led to changes consistent with immune downregulation. A U.S. Phase 1 exploratory study evaluating the effects of helminth infection on MS has shown similar results. In this study, patients with newly diagnosed treatment-naive RRMS were administered Trichoris suis orally every 2 weeks for 3 months. Although the study is ongoing, results for 5 patients showed an improvement in MRI activity even after the well-tolerated treatment was discontinued. Our sanitized world, devoid of many of our old friends, may be the reason for the increase in allergic and autoimmune diseases noted worldwide. Moises Velasquez-Manoff refers to it as the epidemic of absence in his book of the same title. Do we need to bring back old friends? Maybe not. Perhaps it's better to understand the immune system and the mechanisms by which the immune response can be modified. As our understanding of the pathophysiology of MS is ever evolving, so too is our appreciation of the sophisticated immune system and the strategies for controlling the autoimmune response.